Welcome to our video on cell channels and their roles in cellular communication and function. Understanding how these channels operate is essential for comprehending various physiological processes, especially in excitable tissues like nerves and muscles. Local response, also called a graded response, refers to the changes in membrane potential that vary in magnitude based on the strength of the stimulus. These responses are typically seen in dendrites and cell bodies of neurons. Ligand-gated channels open when a signaling molecule, or ligand, binds to the extracellular part of the protein channel. These channels are commonly found in the dendrites and bodies of neurons, playing a crucial role in synaptic transmission and neural signaling. Voltage-gated ion channels respond to changes in the electrical properties of the membrane. They open or close in response to shifts in membrane potential, allowing ions to flow in or out of the cell, which is essential for the generation and propagation of action potentials. Typical resting potential values vary between cell types. In neurons, the resting potential is approximately -70 millivolts. In skeletal muscles, it is about -90 millivolts, and in smooth muscles, it is around -50 millivolts. These values represent the stable membrane potential when cells are not stimulated. The equilibrium potential for specific ions is determined by the Nernst equation. For potassium, it is -96 millivolts, for sodium, it is 60 millivolts, and for chloride, it is -78 millivolts. These potentials are crucial for maintaining the resting membrane potential and for the function of various ion channels. The Nernst equation can be used to calculate the cell potential under non-standard conditions. It incorporates the standard potential, universal gas constant, temperature in Kelvin, ionic charge, and Faraday constant, and is multiplied by the natural logarithm of the reaction quotient. Transmembrane potential refers to the electrical potential difference between the inside and outside of the cell, measured in millivolts. This potential is essential for many cellular processes, including the transmission of nerve impulses and muscle contractions. Muscle cells are controlled and regulated by nerve cells. Nerve cells, or neurons, use electrical impulses to encode, process, and transmit information. Examples include cranial nerves and spinal nerves. Muscle and nerve tissues are called excitable tissues because they respond to external stimuli. This excitability allows them to generate and propagate electrical signals, essential for muscle contractions and neural communication. Protein kinases are enzymes that catalyze the transfer of phosphate groups to proteins, leading to their modification. Examples include cyclin-dependent kinases, which play a role in regulating the cell cycle. G proteins are named for their ability to bind guanine nucleotides, GDP-GTP. They consist of 3 subunits, alpha, beta, and gamma. G proteins are associated with G protein-coupled receptors, GPCRs, on the cell membrane and play a key role in signal transduction. Second messengers are molecules that transmit signals from receptors to target molecules inside the cell. For example, the alpha subunit of a G protein can act as a secondary messenger by traveling along the membrane and attaching to a target, causing a conformational change, such as opening a calcium ion channel. The endocrine system targets cells that are far away, using hormones transported via the blood to reach these cells. This system is involved in regulating various long-term processes, such as metabolism, growth, and reproduction. Understanding these mechanisms is crucial for grasping how cells communicate and respond to their environment, ensuring proper function and coordination within the body. Thank you for watching this video on cell channels and their functions. Stay tuned for more educational content on cellular processes and physiological mechanisms.